Good evening, ladies and gentlemen, and welcome to our new spring series of Science for the Community. My name is Thomas Miller, and I have the honor and privilege of being the director of the Chesapeake Biological Lab, part of the University of Maryland's Center for Environmental Science. The center is Maryland's graduate institution for the environment, and we have a unique mission to conduct research on any aspect of the environment that affects you, the citizens of Maryland, or helps inform people who make decisions on our behalf. So why do we do these lecture series? CBL was founded in Solomons because the community of Solomons came together to build the first lab building for our founding director, Reginald Truitt. And since those days, we have felt a tight bond to our local community. And we constantly seek ways to give back, to tell people about the work that we do. Our mission here is to tell people who we are, what we do, and why it's important. We've been doing these Science for Community lectures now for more than a decade. And in that time, our researchers have presented on a wide diversity of topics, from economics to fisheries management, from the chemistry of rare trace metals to cycles of nitrogen and the ecosystem restoration of the Chesapeake Bay. You can find many of those lectures recorded on our website, and I encourage you to watch them and understand the importance of the work that we do. Each year when we do these lecture series, we pick a theme, and we organized 5 talks around that common theme. And our theme this spring is the urban ocean. As many of you may be following in the news, there is a conference that has just finished trying to find policies that will preserve and sustain the world's oceans for future generations. And indeed, there are some aspects of our oceans that are in peril. We as society impact those oceans most in the coastal zones, and particularly in those parts of the coastal zones that are close to urban areas, such as the Chesapeake Bay, such as perhaps Long Island Sound off of New York, perhaps San Francisco Bay, or Puget Sound off of Washington. And one can think of many such systems around the world where a large urban city is on the coast for the benefits of transportation and shipping and for the benefits of the economy. And it's these dense urban areas that affect the oceans most, and perhaps where we have the greatest responsibility to ensure that the science we have is available for the ocean that we want, and I would argue the ocean that we need. So this spring, we're presenting 5 lectures on the urban ocean. We'll start out with a global view, and I'll have more to say about that when I invite and introduce tonight's speaker. But following tonight, one of our own faculty, Mario Tamburi, will be talking about how we regulate and control the risk of invasive species in the urban ocean. We then have a lecture on metals in the urban ocean and the unique risk of legacy contaminants. The 4th lecture in the series is another of our own faculty who will be talking about the global phenomenon of low oxygen in the coastal ocean, a problem that's particularly relevant for the Chesapeake Bay. And then in the final of the 5 lectures, we take a very different tact, and we've invited a colleague from the architecture school at Morgan State to come and talk about how cities perhaps can plan to help restore and sustain the oceans on which they depend. So it should be an exciting lecture series, and I encourage you to come back every Tuesday night for the next 5 weeks to hear what our researchers have to say. Tonight, it is my pleasure to introduce one of our own, Dr. Judy O'Neill. Judy hails from New England. She got her undergraduate degree at Boston University. She then went on to Stony Brook, and then she came to UMCES, got her degree at UMCES in 1995, and then left for perhaps warmer and sunnier climes in Australia for close to a decade before returning to UMCES, where she has been on the faculty of our sister lab on the Eastern Shore, the Horn Point Laboratory. And part of that global travel that I described in her background led Judy to have connections with a team in Australia that was developing a very ambitious program called the World Harbour Project. And at its core, the World Harbour Project recognizes the important connection between the world's great cities and the oceans on which they depend. And so tonight it is my real pleasure to introduce Dr. Judy O'Neill, who's going to be talking about her work on the World Harbour Project. If you have questions during the presentation, we ask you to type the questions into the chat box. This is a Zoom conference rather than a Zoom meeting. And so we are unable to unmute you, but we do welcome your participation. And at the end of Judy's lecture, I will moderate questions from the chat box to sort of provide a mouthpiece for your questions, and so we can all hear Judy's response. And so with that, as I said, it is my utmost pleasure to introduce my colleague, Dr. Judy O'Neill. Judy, over to you.
Well, thank you so much, Tom, for that great introduction. That was a perfect segue into what I'm going to be talking about tonight. As Tom mentioned, I have been working on the World Harbour Project, which is an interesting conglomeration of people. Here, let me move on to the next. Okay, this is slightly. Sarah or Tom, I don't seem to be up. There we go. Sorry, I didn't think I could have the control, but now I do. Thank you. So tonight I'm going to give you an overview of the World Harbour Project. I'm going to talk about 2 of the working groups that I'm involved in. There are 4, and I'll give you an overview of them. But I'll talk about some water quality in Baltimore Harbor that I've been doing with another colleague, Eric Schott, at our lab at IMET in Baltimore. And I'll also be talking a little bit about our work group 4, an education project in the New York Harbor as part of a Billion Oyster project, using oyster restoration for school curriculum and citizen science. And then I'll just touch on future directives. So the World Harbour Project was the brainchild of this fellow down here, Peter Steinberg, who coincidentally is a University of Maryland graduate. He did his undergrad at College Park, and he is a proud Terp. He has been working for many years at the Sydney Institute of Marine Science in Sydney, in Australia. And there's this growing recognition that the proportion of the global population living in coastal cities is increasing, and it exerts a very high impact on the natural environments, as Tom alluded to in the introduction. And this also gives rise to some social conflicts as well for multiple uses of harbors. So Dr. Steinberg, along with many colleagues around the world, developed this interaction with the goal to develop resilient urban ports and harbors through a global network of collaborating scientists. And this was the launch of the project in 2014 in Sydney with the Minister of the Environment from New South Wales in Australia. So as I mentioned, the World Harbour Project is more of a global research framework. And as I mentioned, about 50% of the world's population lives in cities, and 50% of the population is in coastal regions. And in Australia, which is the Sydney Harbour, where it's the home of this project, Sydney is actually the home of 20% of the entire Australian population. So we think of Australia as a sort of a very rural place, but the cities themselves are very populated. So the idea for this framework is to focus on the importance of the intersection of humans and the urban, marine, and estuarine and environment, and for the need to consider sustainability and management for healthy urban waterways. So this has been a project that started back in 2014. We've had a little bit of a tough go during the global pandemic, but we're trying to get things up and running again. But the World Harbour Project brings together researchers from around the world that are concerned with the health of urbanized waterways and the increasing challenges that are faced. So it's to build this transdisciplinary transboundary partnership to share knowledge on common issues, to discuss strengths and weaknesses that each of the harbors had, to come up with solutions and innovations. And there are 30 partners to date. The red are full partners, and the orangey dots are people who are collaborating just with specific projects. And I just love this graphic, which is a graphic from a shipping, online shipping, an analysis of all the cargo ships and the interconnectivity. And this was from 2012. And it's actually a really interesting site. You can see global commerce in action on this website that's listed down here. But this interconnectivity and this increased commerce between different ports around the world, it drives our economies, but also opens up for environmental vulnerabilities due to introduction of exotic species and other things that you will hear about later on in this series, further in the spring. But anyway, there are 4 working groups that were organized by the World Harbour Project with the core aim to build resilient urban ports and harbors through globally integrated research and management across multidisciplinary boundaries, not just country boundaries, but disciplines boundaries. And so we have 4 working groups. One group works on water and sediment quality, one on green engineering, one on sort of social interaction, multiple uses and users, and the 4th working group is education. And I'm only going to be talking in detail about the 1st and the 4th, but I'll just give a brief overview of all 4 of the working groups. So working group 1 works on water and sediment quality. There's 3 main projects that are ongoing. One investigating contaminants in global harbor sediments, one looking at hydrodynamics and water quality with sediment transport issues in ports and harbors, and one that I've been involved in looking at key microbial groups within harbors related to water quality. The working group 2, which I won't focus on today, is a very important one though, working on how to maintain or restore biodiversity of organisms and habitats in these very built systems where there's artificial structures and global harbors. And their directive is to investigate different materials and designs for the ecological engineering of harbors, and also to make enhancements on some of these surfaces to make not just straight surfaces, but nooks and crannies for different organisms, which is similar to some of the work we're doing over here at Horn Point Lab in Cambridge, where we have some of our researchers are adding oysters to some of these concrete erosion barriers. It's sort of been termed the greening of the gray infrastructure, making it more sustainable. The third working group is multiple uses and users. And as we know, harbors have multiple stakeholders. There's government, business, and industries all might have competing interests. And they have to be managed from a multiple user perspective, from transportation and commercial activities, obviously, that drive the economy of these regions, but also increasingly folks living on the water in residential communities, recreational communities, as well as traditional indigenous population and spiritual activities are focused on the global harbors, and becoming more and more apparent that we need attention to environmental justice issues in these regions as well. So this working group 3 aims to highlight the gaps between harbor users, science, and managers to improve resilience and sustainability, and also reduce conflict. And work group 4 is one that I've been very much involved with, which is education to strengthen links between all the working groups to further partnerships for urban coastal stewardship, to train future scientists, as well as community members and citizen scientists. And we have hosted a series of lectures that are available online from 2017 and 2018 that give background on all the harbor cities. And eventually we would like to institute more global online and graduate courses. And we'd also like to build on some middle school and high school programs, including the Billion Oyster Project that I've been involved in New York, which I will talk about in a little bit more detail. So the first project I'm going to talk about is from our work group 1, water quality, is our Microbial Project. It starts in Baltimore Harbor and has some global comparisons. So human population pressures on our waterways pose unprecedented challenges to our water resources, and sewage contamination is one of those issues. Sewage contamination can diminish ecosystem services and pose a health risk to humans and ecosystem, other organisms in the ecosystem. And traditional approaches for routine sewage monitoring have changed very little over the last 100 years, despite the knowledge that a lot of these culture-based methods where we actually take water and grow up these organisms reflect kind of transient events and capture non-specific sources. These sources that are captured aren't specifically human. They can also be from wildlife. So estuaries in general receive fecal pollution from a myriad of sources. They can come from combined sewage overflow, and it's estimated that close to 4 trillion liters of untreated sewage each year flow into the waterways from overages, even if there are sewer systems during flooding events or storm events. And water that has picked up fecal pollution can obviously contain diverse pathogens, and this can make these bodies of water unsafe for recreational use. And as I mentioned, these traditional indicators to assess human pathogen risks fall into 4 main categories: total coliform, fecal coliform, E. coli, and Enterococcus, which are all just a group of bacteria that are found in warm human and animal mammalian guts. And the standard for the EPA in the US and around the world is the specific bacteria E. coli and Enterococcus. And they set limits for contact standards for water quality at particular levels that are defined as colony forming units per 100 mil of a sample. So for E. coli, the contact standard is 126. For Enterococci, it's 35 colony forming units. So if we take water and filter them onto a specific agar or nutrients for them to grow, each one of these little dots would be a colony forming unit. And so these are very important public health issues because waterborne pathogens can cause between 12 to 16 million cases of gastroenteritis in the US alone each year. And currently, as I mentioned, these fecal indicator organisms such as E. coli or Enterococcus are used to monitor the fecal pollution in the recreational environments, but they are not specific to humans and are abundant in all warm-blooded animals. So they are not actually a good predictor of human pathogen occurrence, nor a very good indicator of pollution source, where the pollution is coming from. So two of the main underlying questions for some of these, for this project, for the Microbial Water Quality Group at World Harbour Project, were what are sources of fecal coliform pollution, and does the standard method of coliform counts reflect human-derived inputs? So the goal was to look at these different sources and potentially develop new indicators of fecal pollution that are more informative than our standard E. coli or Enterococcus, and provide more information about the source, where the pollution is coming from. And much of the work to date had focused on looking at microbial population structure in sewage and comparing with animals. So the project was inspired by one of our partners from Milwaukee, from the University of Wisconsin-Milwaukee, Sandra McClellan, who was developing new approaches for pathogen surveillance using next generation sequencing technologies or molecular DNA work. So she had been characterizing complex microbiomes of humans and animals and had developed new water quality indicators that could potentially provide more information as to the source of sewage pollution. So this was an opportunity to potentially rethink how we measure water quality with these advances in techniques. And just an aside here, this sewage tracking has gained broader use during the COVID-19 pandemic, and our colleague Sandra McClellan has been quite active in that. And the Centers of Disease Control had established this national wastewater surveillance in September 2020, and they've now been surveilling wastewater for COVID in 37 states. And it provides a quick glimpse of whether levels of coronavirus are rising or falling in these hundreds of communities around the country. And it's great because it gives you a glimpse into what might be coming down the road in terms of new variants before people are tested. Because as one of the researchers said, the advantage to wastewater monitoring over testing is everyone has to use the restroom. Not everyone will get tested. So this is just one of the applications of the sewage tracking that has gained broader use during the pandemic. So when researchers started looking at sort of the molecular components of when they started sequencing sewage, they were surprised to find that 80% were of environmental origin, not associated with humans. So 80% of what was being sequenced by 16S RNA was of environmental origin, with only 20% of fecal origin. So folks were sort of surprised by that. So they started looking for indicators that were more, sorry, I just got to move the faces out of the way. Looking for indicators that were more specific to their source. So in 2007, Sandra McClellan and her colleagues started using this next generation sequencing and could take inventories of all the members of a community in the sewage, identified the bacteria to specific hosts using this quantitative polymerase chain reaction, or this qPCR, which has become introduced into everybody's parlance due to the PCR testing for COVID again. So what they could do now, instead of just saying that sewage had E. coli or enterococcus from just anywhere, they could target these different portions and say that it was either from a cow or humans or birds. So this next generation sequencing, these techniques have brought in a whole new era into microbial ecology. And what they found were that they could, this new approach for pathogen surveillance using molecular DNA methods could characterize these very complex interactions between all these different sources of fecal pollution. And they found that several of these sequences, including one for a group of bacteria of Lactospiraceae, these sequences were unique to humans and not found in other animals. So they could start parsing out where the different sewage influx was coming from. So combining these human-associated fecal markers with animal-associated fecal markers can provide a greater resolution and specify where to address the cleanup and mitigation of some of these fecal pollution sources. I don't know why. So to highlight the platform of these new techniques, Sandra and colleagues realized that the World Harbour Project was a great platform to test out these new techniques. So a subset of the 30 countries, 30 cities that are involved, participated in this global sewage survey, and these are the cities that are participated in it over here. And so researchers from all of these countries, these cities, including Baltimore, which included Eric Schott from our UMCES lab in Baltimore and me, all coordinated to sample at 4 dates over a given season at 6 sites and a gradient of human impact. And we ran samples with the old methods as well as with these new molecular methods. So there were 3 key questions for this experiment. Some of them were specific to what the variation in the signals in the harbors were and how those related to environmental factors, with whether they were well-mixed systems, et cetera. Another question was whether the E. coli or the enterococci were a good indicator of sewage contamination, and to do some source tracking about what the major source of contamination in each of the harbors were and how they compare to each other. And some of this work is still ongoing, but I'm going to just be looking at this middle question about whether the old methods, how they compare with the new methods. So in Baltimore, this was our Baltimore crew. Here's Eric Schott. I think you can see my cursor. Eric Schott with the brown shirt in the middle. And the 2 women are our colleagues from Blue Water Baltimore, Barbara Johnson and Alice Vilpita. And in the center, it was my summer student from the University of Puerto Rico, Norberto Arzola. And I also had a graduate student, Morgan Ross, who helped with this project as well. So what we did was we had sites ranging from upstream in Baltimore Harbor and all the way down harbor, and had a control site even out at Sandy Point Beach and some recreational areas at Stony Creek. And we had 3 hypotheses that we were going to be testing. One, whether the fecal indicator bacteria, the FIB, the enterococcus and E. coli, we hypothesized that they would be higher, closer into the harbor, closer to humans, and that they would also be related to salinity and nutrient content, correlating with rainfall and runoff We also hypothesized that the molecular techniques would be better at tracking sewage inputs in general. So we set out, each group in each of the 17 cities set out to collect this data and do the comparison. So here we have the inner harbor, the outer harbor, and then reference or control sites. So for our field methods, we collected water samples in June, July, August, and September. And with that, we also collected water quality parameters, physical and chemical parameters, including temperature, oxygen, salinity, total nitrogen, total phosphorus, and a few other parameters to try and correlate some of the occurrences. And in terms of the lab methods, we filtered the water samples and we grew them up on specific agar for either Enterococcus or E. coli. And we counted those samples to see whether what the abundances were. And this is a pretty standard method. Some techniques have evolved that you don't have to actually grow them and count them under a microscope. They have other techniques measuring things with UV, et cetera. But this is a very standard method around the world and still what is used in most countries. So in terms of hypothesis one, these were the data from our old technique of growing the E. coli and Enterococcus. And here we have the E. coli abundance, and these are our sites from Jones Falls in the inner harbor to the outer harbor and control sites at Sandy Point. At the 4 different times of the year, we measured, collected in June, which is blue, July, which is the orange, August, which is gray, and September was yellow. And what you'll see here is there was a huge variation in the summer. And if you'll recall, I didn't mention it, but this data was taken in 2018. And if you'll recall, the summer of 2018 was the rainiest summer on record. And in fact, I think it was the rainiest year on record for Maryland. So you see this huge jump in the E. coli abundance when we had that increased rain. And you can see it highest inland in the inner bay near Jones Falls and dissipating as we move further away toward the mainstream of the bay. And then the good news is if you swim at Sandy Point Beach, it had dissipated by the time it got to Sandy Point Beach, even with all that heavy rain. The hypothesis too, that the concentrations of E. coli and Enterococcus will correlate with sites of lower salinity. That would be where there's higher rainfall bringing things in, and higher nutrients, nitrogen from runoff or from sewage overflow was also supported. And that's not very surprising because that's pretty much what we would expect. So, but that was a good summer project for our summer students, and it also was the baseline for the comparison for the molecular data. So the results of that, the traditional assessment, were that the E. coli and Enterococci varied on that urban gradient with highest values at most urban, inner urban Baltimore Harbor sites and intermediate values at the outer sites, and the lowest values further down the bay in the least urban sites. And they were lowest with the higher salinity once rainwater was diluted. But the interesting thing was that the indicator bacteria values in the inner and outer harbor exceeded the EPA standards at the majority of the samplings. And I forgot to point that out, that the EPA standard of what's acceptable is this dotted line. So on average for that summer, it was exceeded a great portion of the time. And as I mentioned, there was a significant correlation with the total nitrogen and the rainfall. So then the next part was, how do these results compare to the molecular results from the 7 global cities? So what we did is we took paired samples. So when we were taking the samples for to plate them to grow up our Enterococcus and E. coli, we also filtered samples for the molecular analyses, and we sent them to Sandra McClellan out at the University of Wisconsin, and she employed qPCR methods for those gene markers that had been previously validated as human-specific markers that are listed here, and they are abbreviated LACNO3 and HB for human bacteroides. And so this is a sort of summary of the results, which have not been published yet. Some of these analyses were delayed due to the pandemic. But as I say, the samples were collected from each city at 17 sites around the world, and in parallel with the traditional methods, and the molecular samples were sent frozen to Sandra's lab at the University of Wisconsin. And they were analyzed for those specific gene marker occurrences. And also the microbial community was also characterized. And the goal was to explore the universal applicability of using the human fecal bacteria for global sewage surveillance, and to also help determine the potential sources as well as potential risks of pathogenicity. And so what this interesting visualization shows is a network analysis of 200 of the most common, what we call amplicon sequence variants. And so you can think of these as different kinds of taxonomic units of microbes in the sewage. So each one of these nodes is showing the interrelatedness of these different bacterial communities from the 17 different sites. So what this network analysis shows is where the most shared communities are. So those that are shared around among the 17 cities are the large blue nodes, and the pink nodes are these bacteria that are shared in more than 15 cities, and the green dots are those that are shared in 10 or more cities, and then we go so and down the line, and the gray are those that are shared in more than 12 cities, or less than 12 cities, sorry. So what this analysis tells us, the water samples that came from the 17 different cities demonstrated that indicators specifically associated with the human microbiome were indeed sensitive measures of sewage contamination. And it was found that 40% of the sites tested had evidence of sewage contamination, which illustrated the ubiquitous nature of sanitation concerns across the world, in contrast to the traditional methods that only flagged these sites 10% of the time when compared across all of the sites. And this is another interesting representation, visualization of this data that Sandra and colleagues have put together, showing that the universally present sewage indicators that are specific to humans, this is concentrating on one of them right here, the human bacteroides, and shows it along the 17 different cities in terms of their decreasing abundance of that human bacteroides indicator. So this shows that the city that had the most influence of the human sewage was Rio de Janeiro, followed by Ravenna, Italy, Milwaukee on the Great Lakes in Lake Michigan, in Qingdao in China. And you can see that Baltimore comes up right here, sort of in the middle with Baltimore near Boston, which makes sense as we go along. The ones that had less and less human influence are also harbors that are more well flushed. So that's to be expected. And what we have over here are just those different these different types of indicator species, and this inner number is how many times they co-occurred in one of the cities. And anyway, these visualizations are things that people that are much better at big data than I am at, but I just think it's wonderful that they can that these new programs of network analyses can help us with these big data issues and try and present it in a coherent form. The other interesting thing about Darwin down here is the least impacted by human sewage. In fact, despite having E. coli and Enterococcus, they had no evidence of human sewage in their harbor. So the researchers there now are trying to look at what animal might be dominating the Enterococci and E. coli input into the harbor. And one of the candidates that they're following up on are these large colonies of flying foxes or fruit bats that live in the mangroves around the Darwin Harbor, which I just thought was sort of an interesting little aside. But that just goes to show you that if you just measure Enterococcus and E. coli and you don't know the source, you don't really know how to address the problem. So to summarize the microbial experiment, many of the organisms present in sewage had not been considered as indicator species, but these organisms were found to be universally present at higher abundances than E. coli or Enterococcus across those 17 cities. And we have indeed been relying on 100-year-old techniques, mainly because these are organisms like Enterococcus and E. coli. These bacteria were culturable, or they were growable on these agar plates. But this work has found that a subset of human-specific indicators are consistent across the global harbors that were tested, and collectively these results emphasize that current approaches for tracking sewage are inadequate. And so in terms of future applications, given the risks to human health from pathogenic viruses and bacteria, potential dissemination of antibiotic resistance, as well as economic costs associated with loss of ecosystem services, amenities, or recreational use, there are potentially a lot of benefits to adopting more reliable methods of sewage tracking. And we hope to work with managers and public health officials as part of the World Harbour Project initiative to implement changes. And now that I've touched on, gave you an overview of our microbial project, I just briefly wanted to touch on some of our education projects. So it was interesting when I first was talking to Peter Steinberg about working on this project. He's from New York City, and when I first saw him present this, he didn't have New York City listed, and part of the problem was that he knew too many people to work with in New York, but we had just got funded on the National Science Foundation project to work on education initiatives in New York Harbor. So it ended up being a perfect segue for us to work together. So as you all may or may not know, New York City is one of the largest unified school systems is the largest school system in the United States, with 1.1 million students. And there's not a lot of natural environment around where the schools are located, but New York is an island and it's surrounded by water. So folks at the New York Harbor School and the New York Harbor Foundation wanted to get kids in New York City back in touch with the waterways, with the rich heritage that the Port of New York has brought to the culture of the city. And so they started a school, it's a public school, a magnet school on Governor's Island, just at the base of Park and Brooklyn's over here. And they built a college preparatory school out here to focus on marine and environmental issues. And at the central core of this is the Billion Oyster Project, to use restoration as an education and learning tool. So the basis of the Billion Oyster Project is that they wanted to restore the oysters to the amount that was purported to be there when Henry Hudson sailed into the Hudson River back in the 1600s. The oyster population in New York was decimated by the 1900s with multiple, with the increase in population, oysters were plentiful, and they sustained not only the native populations that were in the region originally, but the early settlers, until they were overfished in the 1880s. So central, as I say, to this Billion Oyster Project as an education piece is that the folks that run this from the New York Harbor Foundation are working with New York school systems with students in STEM-C, which is STEM plus computing, to get educational programs built around these initiatives. So as I mentioned, the goal is to restore pre-European numbers, hence the Billion Oyster Project, and they've trained more than 40 middle school teachers and environmental educators to teach and practice oyster restoration and scientific monitoring to students. So it started at the high school, but the project we had with the National Science Foundation to take some of that curriculum out to the other schools in New York City, in the middle schoolers, to have them create these oyster restoration stations where they could go monitor the oysters. This is all part of, the high school students process the shells that are collected from restaurants, similar to what we do here with the Oyster Partnership in Horn Point here on the Eastern Shore. And they also, as part of the Billion Oyster Project of the school, they have their own oyster hatchery, and they have gotten the oysters from our UMCES hatchery at Horn Point originally. So they have very ambitious restoration goals to, as I said, to get back those billion oysters. And there's a website that we helped create, an educational website that records some of the ecosystem services that have been restored and will be increasing over time. As I said, we had the project that brought more of these education initiatives into the middle school, and we built a curriculum around these issues. And I know I'm running out of time, so I'm just going to highlight a few of these things that we did. We developed a digital platform, and these school groups were able to create their own oyster restoration stations that they visit along the river twice a year, and they measure all sorts of parameters about water quality and oyster growth, and then use that as the basis of some of their STEM studies. I'll just quickly skip over some of that. We've also been working with the Department of Environment there in New York City, and analyzing some of their long-term data to come up with indicators of water quality in the different regions. The students can then compare their water quality with at their different oyster restoration stations as part of their classes with these long-term trends, and come up with sort of report card-like metrics like we have in the Chesapeake Bay. And so just for future directions, we're going to be finishing analyzing some of those, the global comparison and publishing the results, and hoping to work with managers in the future to try and adopt some of these new methods. And we also want to continue some of these education programs and bring some of these programs from New York in terms of the Billionaires Project down to the Chesapeake. And with that, I will just acknowledge my partners at the World Harbour Project, as well as my colleagues at UMCES, and I will leave it at that. Sorry to go over a little bit.
Judy, thank you very, very much. I think this was a wonderful way to kick off a seminar series on the urban ocean, bringing the impacts of society on the water quality, and there at the end as ways to try and chart a different direction and restore some of the function that was there earlier. As I said at the beginning, if you have a question for Judy, don't hesitate to type it in the chat box, and I'm going to try and moderate some of those questions. And I'll start with, I think, a quite broad question from John Overstrom, who says, public health seems to focus on human sewage waste. Does this study reveal that we need to be also just as concerned about non-human contaminants in recreational use of beaches? And I would include, a broader discussion there.
Well, that's a really good question. And part of the impetus for that is that humans, in terms of human health, humans would be more likely susceptible to human pathogens from human sewage. But, there are a lot of unknowns about, the source and its impact on humans, but it also would have impact on the creatures and the ecosystem health in general. So I do think that, contaminants in general are not a good thing, but being able to source track where they come from and then parse out what the impacts are on those different inhabitants of that ecosystem, whether it be humans, fish, other organisms, I think is really important. And I think a lot is unknown about that.
Thank you,
so that's
Judy.
A good question.
Two of the same question in different words from sort of two different people, and they're related to the transition between methods for testing. I think you showed very clearly in the data that the PCR methods are more sensitive, they're more targeted. And so let me just make sure, Ted Karski and David McGowan asks, how do we make the move? Are the new tests more expensive? Is it going to be affordable? How do we improve? We get this higher quality information that the PCR methods obviously provide.
Well, that's a really good question. And part of the reason we've hung on to those techniques for 100 years is that they're pretty cheap. And there will be new costs associated with molecular techniques. But the one good thing about the pandemic, if there could be a good thing about the pandemic, is that the capacity for molecular analysis for PCR testing that came about for increased COVID testing, I think has increased the capacity for molecular analyses in general. However, I mean, that being said, there's still going to be issues finding the funding to do that. But as we found with many of these molecular techniques, as things advance, the costs keep coming down. So this isn't going to be something that changes overnight, but it's something that we need to work towards and figure out how to fund it.
This is a question close to home because Eric Schott has asked it, and normally I try and stay away from our friends and colleagues asking questions, but this is such an important question that I just had to ask it on Eric's behalf. So he makes the notion that many of the attempts to restore the urban ocean have occurred in neighborhoods that are wealthier than most. So how do we deal with the environmental justice issue of ensuring efforts to clean and restore urban oceans are more equitably based than they have been to date?
Such an important question from Eric, because traditionally, these areas have, underserved communities are not the problems, the environmental problems are not being addressed in those regions. And historically, that's where the bigger polluting industries were placed in those neighborhoods. And I think that is something that has to be at the fore of when we do restoration. And I do feel like there has been, a bit of a sea change, no pun intended, that people are much more aware of these social environmental justice issues. And there are a lot of community groups that are coalescing and organizing to really have their voices heard. And even with our local UMCES environmental report card, they are starting to incorporate social and social justice parameters and other economic and social issues into that assessment for our report cards, our environmental report cards for the Chesapeake Bay. And I think bringing more attention to the need for that is really, really important. So thanks for that question, Eric.
And Judy, I'm going to take moderator's privilege and ask a question of my own about the Billion Oyster Project. The whole design of the Governor's Island School and the studies is to bring science to where people are, right? To bring science questions that are relevant for people in their own environments. So you've been doing it for a few years now, and I wonder whether you just have any reflections on how successful and potentially how transformative that approach may or may not be.
Yeah, that's a really good question. And, I think the data's not all in, but the general sense is that when you connect, especially, you got to get the hook in kids in middle school. By high school, they're sort of set. But if you get these middle schoolers all around New York City who are going out to the river or their waterway to experience the environment, this sort of authentic place-based science really does have an impact. It's their neighborhood. They start caring about it. They want to know more. It has, from the data we have so far, it seems to be having an impact. We need some of we have had evaluators and education evaluators in New York City doing this, and we don't have enough long-term data to show how this impacts careers, but we're hoping that the more people get connected with their environment, the more they will care about it. They'll be more apt to be better stewards of the environment and be environmentally literate voters and environmentally literate citizens that will help the community get better.
And I'm just going to ask a follow-on from that, because I've thought about the other side of this coin, is that it is arguably easier to do that kind of very focused education in urban areas simply because the density of young people is so high. Is it transferable to a more rural environment? Many of us, you at Horn Point, we at CBL, are in a more rural environment where it's harder for schools to find the resources to do this very intensive place-based work.
Yeah, it's interesting you should say that, because we're trying to get our environmental education center back at Horn Point back up and running. And we have had a decline over the years in the schools that have been able to get the buses to come to those greater distances on the Eastern Shore. So I do think that it's possible. And I do think that some of these programs you can sort of make them at the smaller scale and have that impact at the school district level as well. And so, Maryland has had some great initiatives, the Maryland Environmental Literacy Partnership, and you know, the fact that Maryland is the first state in the union that required an environmental literacy requirement for high school graduation shows, I think, how progressive Maryland is about the environment and environmental education. And yeah, we've tried actually to get funding for some of these programs, doing the comparison with the rural and the more urban centers. We haven't gotten funded yet, but we haven't given up. So yeah, that's a question we'd really like to follow up on.
All right. Well, on behalf of everyone who attended this evening, Judy, I want to thank you for a really enjoyable and high quality presentation. As a reminder, the lecture was recorded. We will post it to our website in the next day or two. And this is my personal invitation to all of you to join us next Tuesday night when one of the CBL faculty, Mario Tamburi, will be talking about invasive species in the urban environment, in the urban ocean, and efforts to regulate the spread of invasive species. So Judy, once again, thank you very much for a really great presentation. Thank you all for attending, and I hope to see everyone again on Tuesday next week.